Thank you, Dr. Swanson. There are so many variants, it is interesting to consider how these came to be. Today we have Professor Seitz sharing with us to explain how the eye developed evolutionarily. Please explain, Professor Seitz.
I'll be talking about camera lens eyes for you today. Camera lens eyes appear to have evolved during the Cambrian explosion about 542 million years ago. Development of these eyes first started with sheets of photoreceptor proteins which sense light, and these are called eye spots. These eye spots could sense ambient brightness and distinguish light from dark. They also help synchronize the circadian rhythms of the organism. However, the eye spots couldn't distinguish shapes or determine the direction of light. Eye spots have developed over 60 times throughout evolution, so it's not a very rare occurrence for an eye spot to develop. And a modern-day example of this is the euglena. It has an eye spot located at its anterior end, and this allows the euglena to move toward light to facilitate photosynthesis. Over many millions of years, this eye spot gradually depressed into a cup. Now, when light enters this cup, different cells are activated depending on which direction the light is coming from. This allowed organisms to detect brightness from different directions. As the pit deepened, light could be detected in finer and finer details, and this type of eye is seen in snails today. Eventually, as the pit deepened enough, pinhole eyes developed. This is when the pit formed a chamber and reduced the size of the opening to a pinhole, which is what the eye is named after. This allowed for fine directional sensing and even some shape sensing. Organisms first achieved true imaging with these types of eyes. However, they provided poor resolution and dim imaging, but it was still a major improvement over the eye spots of very early organisms. Currently, these pinhole eyes are found in the nautilus.
How did the modern human eye develop to be so different than one similar to snail's eyes?
Well, the modern human eye is actually quite similar to a snail's eye. The only difference is that transparent cells have overgrown the opening, and they've done this to prevent contamination and parasites from entering the chamber of the eye. This allowed the chamber contents in the eye to slowly specialize into a transparent humor, which optimized for color filtering, a higher refractive index, and the blocking of ultraviolet radiation. Let's edit this part out. Okay. We're just gonna edit that part out. Okay. So early lenses, I'm going to restart because I laughed at the beginning of that. Early lenses were composed of epithelial cells, which had a high concentration of the protein crystallin. These transparent cells split into two layers, the cornea and the iris, with a liquid in between. This liquid served as a circulatory fluid to bring oxygen, nutrients, waste, and immune functions to the cells in the eye. The layer also increased the optical power of the eye, which allowed for wider viewing angles and a greater resolution. Essentially, this is the eye that humans have today.
At this point, was the eye capable of color?
The development of color vision occurred fairly early in the development of the eye. It was when receptor molecules were fine-tuned to detect certain wavelengths of light, while most organisms were living in the ocean, and at some point photoreceptor cells developed multiple pigments. This allowed for the detection of different wavelengths of light. This development of color vision allowed for the recognition of predators, food, and other stimuli to the organism.
That's very interesting. Thank you, Professor Seitz. Before these interviews, I did some research of my own and found some very interesting things. For example, the sight of cuttlefish. Cuttlefish view entire polarized light, and the viewing a polarization of light allows cuttlefish many advantages. For example, polarization is used for communication between cuttlefish. Certain parts of the fish, such as its forehead and around its fins, are marked when seen through polarized light, as well as orientation, because it allows the cuttlefish to see through turbulent waters, and for predation, cuttlefish can view translucent prey by polarized light. This highly specified vision is accomplished by specific, though small, variants in the eyes of the cuttlefish. There are high-density streaks of photoreceptors in cuttlefish, squid, and octopi at the approximate horizontal of their environment, which allows for great light sensitivity. Over the photoreceptors are two sets of stacked microvilli called rhabdomeres that are adjacent to each other. Within the microvillar membranes are dichroic photopigment molecules set parallel to the membranes, resulting in greater sensitivity because of maximum directional absorption by the pigment. The filtering of light through the two sets of rhabdomeres facilitates vision of polarized light. Cuttlefish can only see ranges of light from 470 to 500 nanometers, yet are still highly visual creatures. To contrast, human eyes see from a range of 390 to 700 nanometers. Cuttlefish can also pinch their pupils into two smaller mini-pupils, which are then separated into posterior and anterior regions, so the cuttlefish can attack prey from the front and go backward away from a predator. These are the camera-like lenses. I also investigated something interesting about the compound eye of butterflies. Butterfly circadian rhythm specifically responds to ultraviolet light. For example, migrant monarch butterflies travel to Mexico every year and will only fly during the day, specifically when there is UV light. A study was done in which UV light was filtered, and the butterflies lost all sense of direction and started flying in circles. Ultraviolet markings on flowers allow butterflies to land and feed precisely despite their compound eyes, which do not allow for sharp focus. Similar to cuttlefish, butterflies are also marked by specific wavelengths, UV reflective markings on butterflies' wings. There is a specialized dorsal rim first identified in monarch butterflies that is monochromatic for UV light. It only contains ospins that recognize the narrow wavelength that is ultraviolet light. Thank you very much for being with us here today in BioLab. See you next week.